So back to the nervous system. Please go back and review the intro, which was, I did talk a lot about where we're going with the nervous system. So I think it's really important that you go back for the next test if we ever see each other again after all this time off. So we got up to, I'm going to say, about neurons, classifications of neurons, and the functional classifications. Are they motor or sensory? Most of you got that right. For sensory are going from the PNS, where the receptors are, into the CNS. And you should know that the CNS is the brain and spinal cord. The spinal cord is going to do some integration as well. Remember, the interneurons are doing integration. We'll see that in a minute. So if you have a question, just talk out. I can't really see the chat because I'm on this major slideshow here. So it's afferent, right? Afferent is coming into the CNS. Usually anything that's going into a body or like the glomerulus of the kidney, but in this case, it's the CNS, so it's afferent. Now, motor neurons, functionally, are going from the CNS to the target organs. But you remember what I like to call target organs? I like effectors. So muscles, of course, could be smooth muscle, skeletal muscle, or your cardiac muscle. And then you have an association neuron or an interneuron, and these are pretty much all the time are in the brain and spinal cord in the CNS. So these association, and I like to also say integrative. I can't write it over there because this thing's bugging me. Let's see if I can move that first. Integrative. So they're going to integrate how much information or how much depolarization. I think we got this whole depolarization thing down. If not, you got to go back. We're going to talk about it more. What depolarization of this excitable membrane is. Let's see if I can get to the next page. So motor neurons, again, you should also know, which I talked about last time, you should know the difference between somatic and autonomic. So there's a lot of motor neurons in both of these. So the motor neurons, somatic motor neurons, in the somatic part, the effector is skeletal muscles. And just so you know, the neurotransmitter that's going to be released to that skeletal muscle membrane is acetylcholine. Always. And guess what? It's always excitable or excitatory, not inhibitory. It's never inhibitory in the somatic. Right? So that is somatic motor, where the skeletal muscle is the effector in the PNS, of course. Autonomic, now the effectors are your smooth muscle. So let's get this straight. Where is smooth muscle? Number one, smooth muscle is in your blood vessels, mostly arteries. Also in your tubes, walls of the tubes, especially like digestive tubes. And reproductive. And respiratory. So smooth muscle, really important in those areas, and that's targeted by the autonomic nervous system. So cardiac muscle is really kind of simple to break it down. It's not simple at all, but then we're going to do that after nervous system. But cardiac muscle is the heart, just the heart. That's it. Okay, and then the glands, and we're going to learn different glands, sweat glands. We did epithelial tissue. Basically, we knew what the glands secrete, right? And sometimes it's endocrine, sometimes it's exocrine. Endocrine means into the blood. Exocrine means into a duct or out of the body. So I think we're good here. The two branches of the autonomic nervous system, sympathetic, fight or flight. So the neurotransmitter of choice is going to be what's called epinephrine to the effector, right? Now, epinephrine is also known as adrenaline. And I could say norepinephrine and noradrenaline, and it's basically the same thing, and it has the same effect. And the big thing about this is the receptors on the target organ. We're going to talk a lot about that. So the receptors on smooth muscle, cardiac muscle, and glands, most glands, the receptors for the sympathetic nervous system is going to be adrenergic. Listen to that, adrenergic. So the receptors are for epinephrine, adrenaline. It's the same thing. Now the parasympathetic, the neurotransmitter that's going to be released to the effector, which is smooth muscle, cardiac muscle, and glands. Same thing, because this is also autonomic. The neurotransmitter of choice here is acetylcholine. I just write it ACH, same thing. Okay, and rest and digest, or rest and repose. So here's a nice little breakdown here of the functions. So let me just keep that here. So here's your receptors. And this is peripheral nervous system. You can see where that is outside the spinal cord. This is the spinal cord right here. This is not the brain. This is the spinal cord, but it's still central nervous system. So I like to talk about receptors, because without receptors, none of this is going to work. So the stimulus to the receptor, and this is a free nerve ending receptor. And basically in this neuron, and this is going to be classified as a bipolar neuron by the way it looks. So these are actually dendrites. This is true. And this is a sensory neuron. So sensory neurons we know are afferent. So that's incoming into the CNS, into the blue world here, into the ocean of CNS. So free nerve ending like this is most likely for pain or temperature. Maybe tickle. All right, so this is incoming. And then you see the cell body has this one extension, and the axon is basically here. All of this is axon, actually. And most of the time, if not all, these are going to be covered with myelin. And the myelin in the PNS is made by those Schwann cells, which are glial cells. And that makes the conduction a lot faster from the input, that depolarization from node of Ranvier to node of Ranvier into the cell body, where it's integrated again and then brought into the CNS via this afferent. And you should know that all sensory information comes in dorsally. It's not showing you that here. So sensory input, afferent input, comes into the spinal cord through the dorsal root, into the dorsal horn of the gray matter of the spinal cord. And this would be the gray matter, this little under armor H up here. This is white matter out here. So myelinated is the white matter, and unmyelinated cell bodies and interneurons are in the gray matter of the spinal cord. Pretty cool, right? So sensory, and this is somatic. It should also say somatic sensory on this one. That's a somatic sensory neuron, and it's bipolar by what it looks like. So now there could be an interneuron in here. This is where the interneurons are in the lateral horn of the gray matter. And when we do CNS, you'll see that better. So now this is an output now from the, now this is an interneuron between this one. This is synapse here before this somatic motor neuron. All right, so now this is a multipolar, because its dendrites are here. And this is all axon, which of course is going to be myelinated again by the Schwann cells. Schwann cells are the glial cells in the PNS that produce myelin. And these things right here are going to be your axon terminals. And that's where the neurotransmitters are stored in here. Like in this case, in the sensory neuron, they could be here at this axon terminal. So this is a multipolar neuron by the way it looks, right? It has dendrites on one end. I'm sorry, dendrites are here. And the cell body is here. And then you have the axon, which is covered in myelin, in this case. So here's an interneuron, and notice an interneuron is also multipolar. And it's basically what it looks like. So if I ask you what type of neuron, besides sensory and afferent, is a sensory neuron, you're going to say bipolar. Interneurons and motor neurons are multipolar. Hold on, I'm wrong. This is not bipolar. This is unipolar. Sorry. So sensory neurons, and sometimes they call them pseudo unipolar. So I have to backtrack. Sensory neurons are unipolar. Special sensory neurons are bipolar. Correction. Okay. So this is showing now, and again, the motor neuron is efferent. So the motor neuron is efferent, coming from the CNS, going out. Now this is an autonomic pathway here. So this could be either, and it's motor, this could be either sympathetic or parasympathetic, but you'll learn sympathetic tend to have short preganglionic neurons. So you get used to that term. This is autonomic. So this is your preganglionic neuron. It's multipolar. Remember, it's coming out of the CNS. It's motor. It's efferent. So this is your preganglionic neuron, which sometimes is not myelinated. And this is your postganglionic neuron. Just something you need to remember for later in the autonomic nervous system. So right here you have the cell bodies in the PNS. And cell bodies in the PNS are called ganglion. Bundles of cell bodies in the CNS are called nucleus or nuclei. Like right here, this is a cell body of a sensory neuron, and this resides, all of these reside in the dorsal root ganglion, which is another PNS structure that you really need to know later. So remember, preganglionic, postganglionic in the autonomic nervous system neurons. And their effectors are right here, your smooth muscle glands and cardiac muscle. Now here's the axon terminals, these right here. And this is where the neurotransmitters are stored. Now if this is sympathetic neurons or sympathetic postganglionic neuron, they're going to be storing epinephrine or norepinephrine as a neurotransmitter. Not a hormone, this is nervous system. Now the strange part about this, though, if you can remember this is probably the most difficult thing about the autonomic nervous system. So why not talk about it now? You're all high on those great grades you got on today's exam. So this synapse right here, these postganglionic dendrite or membranes are all cholinergic. Cholinergic is receptors for acetylcholine. So all preganglionic neurons in the sympathetic and the parasympathetic nervous system neurons all secrete and store acetylcholine. Doesn't make sense because I told you that acetylcholine was for parasympathetic, but that's only to the effectors. This is in the ganglion. So you have to remember that for later. So let me say this again. In the autonomic nervous system, the motor portion of the autonomic nervous system, all preganglionic neurons store and release acetylcholine. Therefore, the postganglionic neuron membranes have receptors for acetylcholine, and they're called cholinergic. Remember, out here, it's epinephrine to the effector, and these membranes are adrenergic for sympathetic. And again, they'll be cholinergic for parasympathetic. So ACH is what's released, acetylcholine, to the effector. In somatic, always, 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 and it's always excitatory. For the autonomic, it depends if it's parasympathetic or if it's sympathetic. So I'm going to stop and get into your face right now again, if I can get out of here. So listen to this. So here's the deal, right? That's a very tough topic that everybody seems to have a hard time with when you get to the nuts and bolts of it. And it is pretty much nuts and bolts. So think about this. Now, let's just stick with the autonomic nervous system for a second, because the somatic is straightforward. You want to move a muscle, you think about it, that neurotransmitter that's released is acetylcholine. When that signal gets to the muscle, it's acetylcholine in somatic muscle contraction, motor, because it's muscle contraction. I said skeletal muscle. You probably told me a long time ago that skeletal muscle is voluntary. So the skeletal muscle are always going to have cholinergic receptors. What does that mean? What's he talking about? Cholinergic receptors are specific receptors for acetylcholine, which is the neurotransmitter for somatic muscle contraction. And it's always excitatory. It's never inhibitory. So it's always going to get an action potential and make the muscle contract with heavy depolarization. Now, in the autonomic nervous system, you have two different types now. Let me just get out of this thing for a second. So in the autonomic nervous system, you have a preganglionic neuron that comes from a nuclei, actually in the CNS, which you'll learn. So both sympathetic pathways and parasympathetic pathways have preganglionic neurons and postganglionic neurons. Following me? So that's two different neurons in the same pathway. And they meet in the ganglion. Their cell bodies and axon terminals meet in the PNS, which is called the ganglion, autonomic ganglion. And I can go into that forever, but we didn't get there yet. But this is the point. All preganglionic neurons in the parasympathetic and sympathetic, they all release acetylcholine. So they're all cholinergic receptors on all the postganglionic neurons. So now this is why I make such a big deal out of the effector, because the effector is where the work is going to be done, right? So if it's parasympathetic, right? Parasympathetic, the neurotransmitter that's going to be released from the postganglionic neuron is acetylcholine. Always. You with me? Sympathetic, the postganglionic neuron, 99.2% of the time is going to be releasing norepinephrine. Therefore, there's going to be adrenergic receptors on these smooth muscle, cardiac muscle, and glands. Are you with me? There's one exception, I'll tell you later. So now I'm eating my burrito in Central Park again, right? Get ready for this. It's critical thinking. So the bear comes out. You like a bear today? Can we have a bear today? I like bears. So the bear comes out while I'm eating my burrito. So here's the question. What neurotransmitter is going to be released to my small intestine, smooth muscle in my small intestine, when the bear comes out?
Norepinephrine?
It's going to be norepinephrine, I agree. So my smooth muscle of my small intestine has adrenergic receptors. Now here's the next question, Smarty Pants. Is this epinephrine going to be inhibitory or excitatory?
I would hope excitatory.
No, it's going to be
No?
inhibitory. Yeah. Because remember, first of all, you knew that when the bear came, I went from parasympathetic stimulation to sympathetic. So rest and digest would excite my gastrointestinal system, where fight and flight, no. Because my intestines really aren't that important for me to either fight the bear, which you know is not going to happen, or run away. So we're going to stop digesting that burrito. So epinephrine is going to inhibit the smooth muscle in that area. Make sense? So in this case, the point is this epinephrine has the same receptors, adrenergic, but it depends where in the body this is, whether it's going to be excitatory and inhibitory. So
So
see,
if it,
go ahead.
sorry, so while it's inhibiting the parasympathetic stuff like your organs and your digestive system, because the bear came out, you got to make a bolt for it. That would be excitatory to your
Well, what are
motor.
the two other effectors?
I'm sorry,
What's the other two effectors besides smooth muscle?
musculoskeletal.
Mm-mm, not forward anomic. It would be the heart, right?
Oh,
Cardiac muscle. So my
okay.
heart's going to increase. So epinephrine, of course, is going to be excitatory to my electrical system of my heart and my contractility of my heart. And also, here's another, throw another curveball at you. In my blood vessels, especially the peripheral blood vessels, epinephrine is going to cause vasoconstriction, which is going to increase my blood pressure, which helps. What a beautiful dog. So here's another place. What about the respiration? This is a confusing one. What about my breathing? And most of the conductive tubes in my respiratory system that have muscle contraction or muscle innervation is my bronchioles. So when I'm running away from, when I'm in a fight or flight in sympathetic, my bronchioles are going to dilate, right? While I'm eating my burrito, nice and calm, over here in Central Park, they're going to be more constricted. So, because again, I want to get the blood to my intestinal system so I could, get the amino acids from my beans and my rice, whatever's in my burrito, right? So that's a tough concept, and I think it's a good start. At least you know basically what um, adrenergic means, what cholinergic means, along those lines, right? Let's see where we were now. We're here. Okay, so now I'm just going to go into the nuts and bolts, back to the nuts and bolts of the classification of the structure. So sorry about that mistake I made before, because sensory neurons are unipolar. Sensory, pseudo-unipolar, but you could just call them unipolar. And you saw what they look like already. Bipolar neurons are more for your special senses, like retina of your eye, your optic nerve, and the receptors, and also taste, vestibular, hearing, and olfaction. Right? Multipolar neurons are the most common because most, if not all, not I shouldn't say all, but most interneurons are multipolar, and interneurons are the most abundant neuron in your body, in your nervous system. And of course, motor neurons are multipolar, just nuts and bolts. And this is what they look like, remember? So no, that's simple. Okay, and then we have the bundles of axons that's outside the CNS. And these are, I think I went into this before about what a nerve is, like versus a neuron. Like a neuron or many neurons could be in one nerve, and it could be sensory, motor, and autonomic. So we have to get that down. So these are bundles of neurons, which are really axons of the neurons outside the CNS. So they have both sensory, motor. So the nerves are mixed. And then we'll learn the cranial nerves. And most of the cranial nerves, and they're named cranial because they're all basically off of the brain in one way or another. Although you should know that the nerves themselves are peripheral, PNS. The nuclei of the 12 cranial nerves, and they're paired, are in the brain, which you'll see. So some have sensory, but 12 cranial nerves can be sensory, motor, or both, and they can have a lot to do with special sensories input as well. Because, of course, special sensory is all afferent coming in, hence the term sensory, and those are more bipolar neurons. So if you have bundles outside, I mean, you have like highways of axons, and most of them are myelinated. So those are tracks. So like in order for me to move my pen, there's a track of axons that start in of course, it starts in my cerebral cortex and then goes to the spinal cord because it has to descend down my neck, basically, and then out from the nerves of my brachial plexus, which is C5 to T1. So there's a track, and it happens like, let me give you two examples of tracks right now, and see if you could tell me which one's which. Listen to this. Watch this one. There's one track called corticospinal tract. Look at the words, right? Corticospinal. The other one is spino.
Oh, don't like that.
Thalamic. Now, you might not know what the thalamus is, and I'll tell you, the thalamus is part of your brain, upper brain, old brain, deep in below your cerebrum. So, but look what the word spino and think of something. Look at the word corticospinal and think of something. So let me get in your face again and see some critical thinking because the questions are easy, right? So think of those two tracks, corticospinal and spinal thalamic. And they're basically named after where they start and finish. All right. So corticospinal and spinal thalamic. Which one of those two is sensory? Anybody want to play? Think about it. Corticospinal tract. Where does the thought start? What do I keep saying? What's that part of my brain that starts or initiates a thought? Or I should, yeah, it's a cortex, right? It's a cortex, the cerebral cortex. So in this case, it's the signal is starting in the cerebral cortex, cortico, and then going down to the spinal cord. So that's a motor tract. That's my somatic motor tract, so which basically helps me write or helps me kick a soccer ball, right? Now, spinal thalamic comes from the spinal cord up to the thalamus. The thalamus is a relay station for all sensory input except for smell, right? So all sensory input comes in from, goes to the cerebral cortex for cognition. Now, cerebral cortex is not only for motor, it's of course cognition. I may have said that. So our cerebral cortex, that gray matter, that's 7 or so millimeters of gray matter in the outer portion of our cerebrum, is not just motor, voluntary motor. It's also cognitive sensory. So I know what I feel. So the spinal thalamic tract will tell me if I step on a nail, right? I'll cognate that through the lateral spinal thalamic tract, which is specific for pain. Lateral spinal thalamic. So that's a tract that's heavily myelinated because I got to find out about that quick, right? Of course, I have a reflex to get away from the nail, but that's another story. But at least I have to know I stepped on a nail. And that would be spinal thalamic. Afferent coming from the PNS to the CNS, ending up in the cerebral cortex, the primary somatic sensory area of my cerebral cortex. Now, if I want to write with a pen, that starts in my cerebral cortex, the primary motor area of my cerebral cortex. It happens if I'm right-handed, which I am, that signal starts on my left side in the primary somatic motor area. And it crosses over, decussates. The word for crossover is decussation. So that is voluntary somatic motor, and that's corticospinal tract. Make sense? You're getting terms down, nuts and bolts. I'll figure this stuff out. Neurology made easy. Let's see if I can go back to that hell slideshow I was doing before. It's hell, I tell you. So very good, very excellent job. So now we'll talk about the glial cells. These are interesting. I might have mentioned some of these before. I know I mentioned the Schwann cell because it's got a cool name, and it's in the PNS, and it's always all textbooks use the Schwann cell as like that first glial cell they introduce you to. It's also called the neurolemmacytes. But I'm so old, I learned this on a cave wall. I never even heard of that until like 10 years ago. So Schwann cells are producing that lipoprotein myelin that makes white matter white. All right, so tracks, of course, have many axon fibers that are myelinated for speed, organization, right? So now we're talking about the PNS only, and there's only two. There's the Schwann cell, which I think we met before, and then we have these satellite cells. And these are called, all of these are going to be called glial cells, and they're not neurons, they're supportive cells. I think I might have put a question on the exam today, I'm not sure, that about neurons not really able to undergo mitosis because they have less centrosome. But glial cells are very mitotic. Glial cells have nucleus. They undergo mitosis pretty rapidly too. So you should know that about glial cells. And they're not neurons, they're supportive cells. And they don't conduct any electricity. They do have, like Schwann cells do have a membrane that, where that signal jumps from node of Ranvier to node of Ranvier called the neurolemma. And you'll see that. So satellite cells, I really don't know all that much about satellite cells. I do know that they may act as stem cells and they may help regenerate neurons that are injured, and they may have some phagocytic function. So we'll stick with like the more like stem cells as a glial cell, ready to undergo mitosis and differentiate into a supportive cell that we really don't know that much about. So there's a lot to be learned in the nervous system. So that's two that are in the PNS. I should know the names. Now the CNS. Now the myelin-producing glial cell of the CNS in the brain and spinal cord is called oligodendrocyte. So I guess that's like the cousin of the Schwann cell in the CNS. Right, so it produces myelin. They look different though. They look different. These have more like extensions, like foot-like processes that wrap around the neurons in the brain and spinal cord. So there's four in the glial cells in the CNS. The second one is we're going to call microglia, and these are basically the garbage man, basically phagocytize, which is an immune response, engulfs foreign substances and degenerated material like proteins, like faulty proteins. Because again, we talked about genetics, how you can have slight mutations all the time, and this excess or faulty DNA or RNA really and proteins like cytoskeleton proteins need to be cleaned up, right? To keep that central nervous system, clean and working properly with all its synapses. Astrocytes, star-like cells, glial cell of the CNS, very important. These regulate the external environment, but more important, astrocytes are involved in what we call the blood-brain barrier. So the astrocytes are intimately related with the capillaries of the brain. So it's very picky. The brain is very picky. These astrocytes are very picky what they allow into the neurons for good reason. Of course, neurons, they want to just use glucose. And it's kind of strange. It's kind of weird evolution, but the brain neurons, the brain cells basically only want lipid-soluble substances to come in. But glucose, we know, is polar. It's not lipid-soluble, but magically it gets in with the help of insulin, of course. Oxygen, very lipid-soluble. Thyroid hormone, lipid-soluble. Carbon dioxide, very lipid-soluble. So this is a very selective, tight junction cells. Even the capillaries of the brain have very tight connections to each other, the actual endothelial tissue. It's not porous like in our lungs or in our tissue. That's simple squamous epithelium. So this tightens up that blood-brain barrier. So there's the astrocytes. Think blood vessels. Think the connection of blood vessels. And then we have ependymal cells. This is the last one. We're almost there. Ependymal cells secrete CSF, cerebrospinal fluid. And this is more for protection, cushioning of the nerve tissue, the brain and spinal cord. Of course, this is CNS, remember, or is it CNS glial cells. So ependymal cells line what's called ventricles, and ventricles are spaces in the brain, that are filled with CSF. And you'll see that when we do the CNS. Ependymal cells form structures called choroid plexus. And I believe there's 4 of them. So choroid plexus is a structure made mostly of ependymal cells that secrete the cerebrospinal fluid into the ventricles, which are the spaces in the brain. So the blood comes in, and then in this choroid plexus, the blood is converted to CSF, which is kind of a lipid carrying substance. It has some sodium, a lot of water, some white blood cells, very little glucose, because it's really just a cushion and bathe and provide some nutrients to the cells, the brain cells, the neurons, right? So ependymal cells are more involved in the blood, listen to this, confuse you even more, blood CSF barrier. What gets into the CSF? So a lot can go wrong here, right? A lot can go wrong. And you'll see. So here's the CNS. Here's an astrocyte, kind of looks like a star. And see, this is a blood vessel, capillary, tight junctions, blood-brain barrier. Right? Here's another good one. Here's our friend, the oligodendrocyte, the friendly cousin of a Schwann cell. Doesn't look anything like a Schwann cell, right? It produces myelin, which wraps around these axons to speed up conduction, right? Saltatory conduction, jumping from node to node, speeds it up. Thanks to the myelin, the white matter. The cell bodies are all gray matter. The gray matter, like if this was my cerebral cortex, it would be all cell bodies and some dendrites. Most of the axons are myelinated in these particular fibers. Here's the garbage man, microglial, right? It's kind of more of a phagocytic cell. It looks dendritic, doesn't it? With all those arms and tree branches. So hopefully if there's some excess proteins being transcribed and then translated on the ribosomes, and it's kind of faulty, like something actually can cause something like Alzheimer's, right? Or dementia when you have too much beta amyloid proteins that are genetically cleaved at the wrong place and they become insoluble. So hopefully the microglial cells can clean up some of that excess plaque-making proteins, besides bacteria and virus and all, and whatever else faulty RNA is in that intercellular space. And look at the perivascular feet, right? And you know, the oligodendrocytes have little feet processes too. Sometimes they call them pedicels. Right, so you see those extensions. And then you have ependymal cells. Ependymal cells look, again, very tight junction. They look like cuboidal epithelial cells. And they have some villi, or these are actually cilia, that kind of moves the CSF around. And that it does have a flow, you know, through, and it kind of gets recycled. And there's another place where it's recycled back into the blood, which is a whole nother thing we learn in CNS. I always like these. Basically give you a little breakdown. Look, all 6. All right, so here's the PNS ones above the line, and there's CNS below. And you have a good idea of what they all do. So you met everybody. You met the whole family. You have the neurons, which is what's called parenchyma. Parenchyma is the functional cell of an organ. Like in the kidney, you have what's called a nephron. Nephron is the functional cell of the kidneys. So the stroma, is more connective tissue, like reticular connective tissue that kind of holds things together, like organs and brains and other parts. Glial cells are part of the nervous system, just that they're not conductive. And there's many more glial cells than there are neurons, believe it or not. Now, of course, because neurons are amitotic, which means they don't undergo mitosis. Basically, once your brain cells and your spinal cord and your nerves, peripheral nerves develop, that's it. That's what you have. And that's what you're going to have for the rest of your life. Where these things are all mitotic, they're always turning over. So here's a question, which one is more likely to have a tumor? Tumor is excess mitosis. Of course, it's going to be the glial cells. So most CNS tumors are not tumors of the neurons, the parenchyma. Most of the tumors are glial cell tumors, like astrocytoma, ependymoma. I think I just made that word up. Right? So, or a glioma is really the word they use a lot. Glioma. And it's rapidly growing. So even though it doesn't sound, well, that's not so bad. The problem is if you have excess growth of these glial cells, you're going to have space occupying neoplasms. You're going to have, your ventricles are going to get crushed. Your brain tissue is going to get crushed. The vascularity, the blood coming to and from the brain is going to be compromised because it takes up space and they grow so rapidly. And remember I told you the CNS or the brain has a blood-brain barrier? It's really hard to get medications through that blood-brain barrier, especially if they're not lipid soluble. Like antibiotics, for instance. Most antibiotics are non-lipid soluble. You can't get those into the brain if you have an infection, an encephalitis. Some people get encephalitis from Lyme disease, right, which is a bacteria that gets into your brain, gets into the CSF, gets into the blood vessels, gets into the blood-brain barrier. But the antibiotics, which is supposed to kill the bacteria, stop it from going through mitosis, can't get into the brain. So sometimes you have to do a shunt. You have to circumvent the blood-brain barrier by putting a different type. It's almost like putting a pipe from the lower part of your brain into the higher parts of your brain. Depends on where the infection is. So that's not such nuts and bolts. Myelin sheath, we already met the myelin sheath, right? All axons in the PNS have that because we want speed. We want to know if I stepped on a nail. We want to, get that pen to the paper, right? Of course, while buying a new car or something like that. Sign and drive. Got to do that quick. Get out of there before they change their minds, right? This is what I mentioned before. So outside the Schwann cell membrane itself is not conductive, but the outside of the Schwann cell makes a nice surface for conduction over it. So the depolarization jumps over the neurolemma. So the outside of the Schwann cells along these axons is called neurolemma, and the part or the membrane around the axon itself is called an axolemma. So if you don't have myelin, you're just going to have an axolemma, and it's going to be slower conduction. If you have Schwann cells and myelin, either way, PNS or CNS, the neurolemma is going to speed up the conduction. There you go. Okay, so they wrap around. You've seen that. I showed you the myelin sheath in the PNS, which is basically Schwann cells, made by the Schwann cells, maintained by the Schwann cells. And there's gaps between the Schwann cells. And again, they called it something fancier before, but I like just to say node of Ranvier. And they're unmyelinated parts. And this is where most of the sodium channels are. Because once the sodium channels open, and potassium too, once the sodium channels open and sodium comes into the cell, that's depolarization. So each time that conduction, that depolarization flies over that neurolemma to the node of Ranvier, it opens up another channel, then another channel. And of course, it has to repolarize, and that's when potassium exits. So depolarization, sodium influx, repolarization, potassium efflux. So this is dipole. I can't say this enough. This is so important. And this is repol. So all neurons have to repolarize after action potential in order to be able to receive another stimulus in case you need another one. So this is kind of cool, just showing you, this is the axon. And this could be motor or sensory. It's just an axon. Here's the axon here, and then the sheath forms this lamellated sheath as it keeps producing and wrapping. And this is in the PNS. That's why it's called the Schwann cell. And this would be part of a node of Ranvier here, where the sodium channels are. And they have nuclei. You can see the Schwann cell nucleus, because neurons have their nuclei in the cell bodies, right? Perikaryon, sometimes it's called. Again, continuing. And, it's more rapid, basically. That's the takeaway of a conduction, right? Here's some electron microscopy of the actual myelination. High power, right? PNS, you see Schwann cell. Okay, now in the CNS, you have oligodendrocytes and you have extensions, same thing, right? Same thing. It's a little different because you don't have the neurolemma. We don't call it a neurolemma in the CNS, but we can say axolemma. And remember this, I already told you that, where did we go here? Myelin. White matter is myelinated, and gray matter is mostly cell bodies, which are never myelinated. And dendrites, same thing. So the myelination is around the axons, and that's the white matter. Tracks, too. Tracks. Like the corticospinal tract and the spinal thalamic tract. And most of the neurons in nerves, peripheral nerves, like spinal nerves, are also myelinated, the axons, within the bundles of nerves. So this is more of a CNS cell. It looks a little different. You have more, and this is kind of introducing you to circuits, like the way there's different circuits. Like it's like wiring going to and from different parts within the CNS. Like remember I mentioned the thalamus, right? The thalamus is a relay station for all sensory information except for smell. So that's incoming. So the next stop after the thalamus is going to be the cerebral cortex. All right, so there's circuits between other parts of the brain and the thalamus, and then to the cerebral cortex, right, for what we cognate. Because, like, again, everything except for smell. But, like if I step on a nail, I might say, wow, I remember that. I stepped on a nail like 20 years ago, and now I just remembered it. So some of the memory parts of your brain, like your hippocampus or your frontal parietal lobe, will get involved in this circuit. Simply stepping on a nail, it could remind you of something. Why? Remind you when I went to summer camp. I never went to summer camp, but it sounds cool. When I was in summer camp, I had a splinter, and it just comes back because of that circuitry. And this is how something like post-traumatic stress syndrome actually happens. This involves an area of your brain called the limbic system, which is very more of an emotional area of the CNS, which we'll talk about. But there's all connections, is the point. Circuits. And there has to be myelination so these circuits could act very quickly. But not all CNS interneurons are myelinated. A lot of them aren't, actually, especially in the spinal cord, which you see. Right? So you have problems with myelination. Right? The most obvious is multiple sclerosis, which is a CNS condition. And most likely, it's from a autoimmune etiology. Etiology means the cause, where your lymphocytes are actually destroying the myelin-producing cells like Schwann cells and oligodendrocytes in the CNS. So it affects the PNS, right? Causes demyelination, right? So if you have less myelin, you're going to have less muscle contraction or slower muscle contraction. And for some reason, this affects females more, affects the muscles of the eyes and the legs, right? So trouble walking. And it can become progressively worse. And ultimately, the problem with any of these neurological conditions is possible paralysis of the diaphragm, right? If the diaphragm controls your breathing without thinking about it, the contraction and distension of the diaphragm has to do that on its own. So most people with Alzheimer's or ALS or severe multiple sclerosis wound up succumbing to respiratory problems because of the issue with the diaphragm. So they'll be put on a ventilator ultimately. horrible condition and there's really no cure for any of those three that I just mentioned. Guillain-Barré is basically another autoimmune condition, but it could happen from a virus too, or sometimes it happens for no reason. So your immune system just temporarily attacks, right? It's a rapid onset of muscle weakness. Sometimes people get this after surgery, but it's pretty much self-limiting. Like this technically is a syndrome, not a condition. And it could affect the CNS and the PNS. So Guillain-Barré is basically the same etiology as multiple sclerosis, meaning your lymphoid cells, your T cells, your white blood cells are attacking the myelin sheaths. So keep that in mind. This is, and your neurons, if they're cut, let's say they're severed, the neurons themselves can't really regenerate because they don't undergo mitosis. So in the PNS, the Schwann cells get involved, and I believe the satellite cells get involved as well in repairing that, let's say that axon, right? If an axon is injured, picture that, like even though nerves are bundles of axons for motor or sensory. So if you have a nerve injury, you could irritate both the motor axons and the sensory axons. So you could have weakness, paralysis, like flaccid paralysis slightly, or depends on how bad the injury is, and you could have numbness with no feeling, or hypersensitivity which is pain or burning. So the neurons themselves don't regenerate, but the Schwann cells form like a tube, a neural tube. Actually, it looks like a kind of like an inner tube that kind of grows together and helps the conduction continue post-injury. So hopefully that neuron will start to connect a little bit better. I guess it's like if you have a frayed wire, and the wire is the axon, and if you put like electrical tape over it, and the electrical tape would be the Schwann cell, or mostly Schwann cell. This is PNS mostly. Can't really talk about that in the CNS with the oligodendrocytes. So it forms like a tube, like the tape will form a tube, right? So again, if you could have, a problem, now this is CNS. Now CNS is a whole different story. If you have, death of a receptor, if you have some toxicity that destroys the dendrites, which is really the receptors in the brain, then you might undergo apoptosis, which is a cell death of oligodendrocytes. So you're going to have where the myelin is, it may stop working. So again, this is not exactly what ALS is, but the toxicity is. Toxicity can cause these death of these glial cells and ultimately the inability to conduct along these axons. Okay. So inhibitory proteins in the myelin sheath prevents regeneration. So the brain is much less regenerative than the PNS axons. Yeah. So again, very picky, and it's got to be protected, right? The brain's all these brain injuries, these concussions, and encephalitis, traumatic brain, TBI, traumatic brain injury, are really cumulative. Once you're injured once, and soccer player whacks their heads together, going for a header, one concussion is cumulative to a second concussion, adds up. Third concussion, you should never play soccer again because you're risking loss of brain function and possible MCI, mild cognitive impairment. So it's not regenerative. All right. So this just shows you how the neural tube is formed ultimately in the PNS, though. This is sorry, I'm blocking your screen. This is in the PNS where you have the Schwann cells. So if it's injured, of course, it takes time. It might take some months of physiotherapy to get these faculties back so you could move the muscle the way you were before. And sometimes you might not get it all back because these neurons, these axons really don't regenerate. It's just the growth around this Schwann cell. So this is like the electrical tape. that the Schwann cells provide to try to keep that motor neuron conducting. So now at least the neural lemma is back in place, and maybe we can get from node to node across that neural lemma and get that acetylcholine released to that muscle and be able to write again when these faculties are lost. So I mentioned the term plasticity last time, and I was referring more to the CNS, the brain. And it's not the plasticity is not growing neurons or repairing neurons. Basically, it's forming new pathways for memory, maybe new pathways for function. Because again, remember, if you're moving a skeletal muscle, where does the signal start? It starts in your cerebral cortex. And your brain is mapped out for every part of your body to move a skeletal muscle. So you could overcome by some rehabilitation or physiotherapy or occupational therapy over time and maybe get some of those faculties back because of something like plasticity. We'll talk about cerebrovascular accidents and strokes. I might have mentioned that last time. But that's PNS is much more regenerative. Okay. So again, you can stimulate growth of collaterals, and it's really not there yet as far as, can we regrow neurons? So you're just forming collateral synapses, really, right? Proteins, NOGO, NOGO. And this is produced within the oligodendrocytes, are actually inhibitory to regeneration. It stops the regeneration. So a lot of research with those proteins. Again, so you'd have to, knock out a mice's no-go, right? Knock the mice out or knock that gene for that protein out and see if the neurons will regenerate if injured. So a lot of research going on with these stem cells, right? Stem cell therapies. Putting in new cells that do undergo mitosis. So there's one place in the brain that we might have some regenerating axons and neurons, and that's the hippocampus. So in certain, like the hummingbird, that has a very highly mitotic hippocampus for some reason. And they will, use those type of cells to maybe regenerate some of those cerebral nerves in humans ultimately to treat things like TBI, traumatic brain injury, concussion, dementia, Parkinson's, all those primary neurological conditions. So neutrophins promote neuronal growth in fetal brain, right? In a developing brain. So we're talking about, post-stem cells. And if we could, use these growth factors that we find in these things like the hummingbird or in a newborn, we can use brain-derived neurotrophic, that means growth, neurotrophic factors. So these are all in a developing brain, but not in the adult brain, although we can isolate some of them. Glial-derived neurotrophic factor, all basically doing the same thing. Neurotrophin 3 and neurotrophin 4 and 5. So think of the word neuro and trophin when you're studying these. Trophin means to produce or grow, tropic, trophin in this case. Right? So that we do have neurotrophins as an adult too, but that's for maintaining your ganglia. Very important, so I can run away from the bear as an adult, right? I have that autonomic faculty because I'm not fighting him. So here's your most abundant glial cell of all 6. And we've seen those interactions between, remember, when you hear astrocyte, think between brain cells, neurons, and blood. Because this is CNS, right? This is CNS. And this is mostly in the brain. There it is. And again, here we're getting, now we're in the brain, right? And this is a good picture because you could see the astrocyte, the connection here between the blood and what's in the blood and what gets out. Now, these are simple squamous epithelium, not very porous, though. It's fairly tight junctions in here, as opposed to in the tissue in the lungs. So in the neurons, this is the axon terminal, right? Axon terminal. And this is where your neurotransmitters are stored. So this is the brain. And there's a neurotransmitter in most of your brain cells called glutamate. It's actually an amino acid. Stored in vesicles. So neurotransmitters are stored in vesicles in the axon terminals. Now, if this axon is excited, of course, or if something gets in that excites it, it's going to release that neurotransmitter into the synaptic cleft, which is the space between two neurons. So this is your postsynaptic cell, and it has receptors specific for glutamate. Now, glutamate in the CNS, and I shouldn't make a broad term here, but in your brain neurons, I'll say it this way. In most of your brain neurons, most of them, glutamate is always excitatory. It's going to cause an action potential. It's going to cause depolarization of the postsynaptic membrane, which is here, this neuron. So these are basically sodium channels. These receptors are tied to sodium channels. The glutamate never gets into the cell. The glutamate only binds to the receptor and then causes depolarization, which is sodium coming into the cell. And that excites the next membrane of the postsynaptic cell. So glutamate, I could go ahead and say, is the most excitatory neurotransmitter in the brain. Right? We might have talked about anxiety before. And the most inhibitory neurotransmitter in the brain is called GABA, gamma aminobutyric acid. And that's inhibitory. Does not allow an action potential. Where glutamate is excitatory. This is inhibitory, well, negative. This is excitatory. Let me just go to the next page before I get in your face. Yeah, let's just talk about glutamate for a second, because I think that's important. Now we just step away a second and just see what we're talking about here. So last time we talked about anxiety a little bit. Did we mention that? Yeah. So again, in the brain, if you're getting, glutamate release, it's causing depolarization. It's causing action potential, which is completely appropriate. And it's released almost as much as acetylcholine is in the body. So somebody who has a generalized anxiety disorder might be releasing too much glutamate, and there's too much connection of glutamate to the receptors. So somebody who has anxiety is hypervigilant, right? They're always worried. They're not sleeping as good as they should, right? So that would be an increase in excitation, right? Which can get out of control if it's happening over and over. So, or the other side of that coin is there's not enough GABA being released from those axon terminals. So your GABA levels are low. Because GABA is inhibitory, right? It'll keep you smoother, right? It'll keep you sleeping. It'll keep you maybe more being able to concentrate a little bit more, right? Without being distracted by other things because you're hypervigilant. So, when you use medications for this, like what's the medication we use for anxiety? Did we talk about this last week? I don't remember. Yeah, those benzodiazepines, right? Or Xanax, those kind of things. They're GABA agonists, which mean they'll help bind to the GABA receptors because the chemical is basically an analog for GABA, and it will inhibit action potentials. Therefore, everything is cool. You're not worried.
What about beta blockers? Does that do the same thing?
No. Beta blockers are for adrenergic receptors, like for epinephrine. So beta adrenergic receptors, like in your heart, you have beta 1 adrenergic receptors. In your blood vessels, you have alpha alpha 1 and 2 beta adrenergic receptors. So when epinephrine is released and it binds to those receptors, it's going to speed up your heart rate. It's going to contract your blood vessel walls, smooth muscle, and cause your blood pressure to increase and your heart rate to increase. You follow me? That's important. And then some people can't afford that. Some people have a weak heart. So you don't want to stress the heart. You don't want to put demand on a heart that's already failing, because then it'll demand more oxygen and it'll strain more and more likely to have a cardiac arrest or ischemia, which is lack of blood flow to the cardiac muscle, to the cardiac muscle, heart muscle, myocardium. So what beta blockers do is block the receptor, so your heart beats slower, your blood pressure goes down. So most old folks, you can look in your grandma's medicine cabinet. I guarantee you're going to find the beta blocker in 80% of grandmas.
Why do a lot of performers use it?
Beta blockers, probably to calm them down, to lower the tachycardia.
I have a question.
The only problem with that, if you're using it as a dancer, think about this. If you're using a beta blocker as a dancer, the beta blocker ultimately does is stop calcium being released into your cytoplasms, which could decrease your muscle contraction and throw off your calcium homeostasis. So I would say a beta blocker is more to calm them down, slow down their heart rate, so they can take their time a little bit more, and not the same way taking Xanax would do it. You know what I mean? There wouldn't be so many side effects. So yeah, and think about somebody who plays golf. They do the same thing. They take beta blockers to perform better, and calm them down that way by physiologically slowing things down. It's not a psychological medication. So that's the best I could say about why a performer would do that. Yeah, but it also may affect them ultimately, because every drug has a side effect, Sometimes as a performer, you need to have fight or flight. You need to have a sympathetic reaction, right? It's adrenaline. Sometimes you work on adrenaline, which is a positive thing. It's completely appropriate, don't you think? You're looking for that E minor or F sharp major diminished ninth chord, and you didn't practice it that much. I bet you when you're in fight or flight, it comes back to you easier. Don't you agree? Like you ever drive in a fog, and you never, of course we don't live in a foggy area unless you go up the Palisade Interstate Parkway. But all of a sudden you remember, I remember where my fog light button is now. Well, you wouldn't remember that unless you had a sympathetic. So sympathetic is a good thing. It's just got to be short term. Like during a performance, right? So why block the sympathetic, I say, Or just roll a big fat joint and smoke that before you go out and perform, like my eras did. Who knows? I don't know. My performance days are less these days. So. Okay. Any more questions?
First, I have a question.
Yeah, go ahead.
I was wondering what would cause low levels in GABA.
What would cause it?
Yeah.
Well, they're all made. And think about the enzymes, right? What if you have a faulty enzyme that produces GABA, right? Or releases GABA. So most of the time, that would be genetic, your body chemistry, right? On top of it. So that it's all chemical. It's all chemical. So like low levels or high levels of glutamate. I don't know why, but like somebody with ALS, amyotrophic lateral sclerosis, if they have a specific gene on the 17th chromosome that happens to stop the suppression of glutamate, so it's too much glutamate and it's toxic. So some of it could be some genetic transcription with those neurotransmitters. But that's a really good question. It's a really good question. Why does, like, under the same circumstances, of course, if you have identical twins and one of the twins has anxiety and the other does not, and everything else environmentally is the same. So that's still a good question, I think. So what external events could affect it? Your limbic system, right? Your limbic system. We didn't really talk about that yet, but your limbic system is basically has the amygdala and the cingulate gyrus, with the hippocampus and this hypothalamus. So all those connections, there's so many connections and different neurotransmitters releasing. Sometimes the cocktail doesn't work. And then you have memory, like post-traumatic things that probably could ultimately affect your release of glutamate and how much glutamate you're storing, right? So again, we're finding more about that, but I tell you, we don't know that much about the brain overall. So all those questions are good. Anything else before we go into the nuts and bolts of astrocytes? I think we're going to next. Yeah, it's pretty interesting. The twin studies, like that. Maybe Claire has a twin somewhere, who knows? So astrocytes, and this, we're talking about potassium here. So they take up potassium from ECF, right? ECF to maintain the ionic environment. It's really important because you want to keep potassium in the cell to keep that membrane potential going, right? Sometimes it leaks out to keep the resting membrane potential, right? They take up extra neurotransmitter. So here's another answer to Jessophat's question. The condition of the astrocyte, right? The connection between the blood, right, and how glutamate is recycled and the enzymes that are affecting the construction of glutamate or the breakup of glutamate or the reuptake of glutamate. So there's a lot of different things that are important. And we looked at astrocytes. I think we got a good idea. They can store glycogen. That's interesting. And they produce lactate. But let me tell you something. We don't want to use lactate in our brain or our heart. We want to maybe convert it from our liver and get it back to pyruvic acid, but we don't want to really have anaerobic conditions in your brain or your heart, right? So the glycogen is there as a backup, and it's a really amazing system. And we learned all the respiration issues. So again, no, they're needed for synapses in the CNS. Of course they are. They regulate neurogenesis, right? This helps with plasticity. So I'm going to write that in there. And this is the most important for us, learning about the blood-brain barrier as it involves astrocytes. And release of transmitter molecules, of course. And these are all neurotransmitters. We'll go into each one of these, and there's so many neurotransmitters, but we're going to do the main ones. Okay, so now I just want you to know glutamate. So we do the CNS overall. Again, calcium, important for nerve conduction and muscle contraction, which you're going to see in the cardio, but also for nerve conduction, because calcium is the mineral needed for neurotransmitter release. Remember, the neurotransmitters are stored in vesicles in the synaptic terminals. So you need to get the calcium into the cell, and intra means in. So astrocytes have a lot to do with that concentration gradient, right? So calcium, really important in the nerve and muscle, right? And release of prostaglandin E. And this is a hormone in the blood. And prostaglandin, some places cause vasoconstriction and sometimes causes vasodilation. So in the brain, this prostaglandin specifically is more about vasodilation, increasing blood flow, which is really important in the brain because it's about oxygen. So this is more vasodilation, but most of the time prostaglandins in the rest of your body pretty much are released in inflammation and cause in tissue, it can cause vasodilation, but also a rebound vasoconstriction, which can cause pain. So prostaglandins are a big source of pain chemical in your body, but they also have a lot of important functions, and this is more endocrine. So we'll revisit prostaglandin, but remember it has something to do with astrocytes because astrocytes are involved with the blood. Prostaglandin is a chemical release from the cells into the blood. So we'll revisit the prostaglandin more when we go into endocrine. So this is less porous, like the capillaries have tight junctions instead of the porous junctions. This is astrocyte up there. So it's selective, right? Very selective on what it lets through. And those epithelial cells are the blood vessel cells. And of course, you know, the transport systems of the membrane are a little different, a little more selective. So all cells or membranes are selectively permeable, but in the brain, they're especially tight, especially impermeable, really, or less than semi-permeable. So again, they can produce ion channels and enzymes that actually destroy toxic substances. So it has some type of immune effect, right? And this is what I was mentioning before. We have problems getting antibiotics or chemotherapies or immunosuppressant drugs into the brain because they can't penetrate. Because the blood-brain barrier is not permeable to any water-soluble substances except for glucose and insulin. So the neurons, the brain cells, are really all about using glucose. They don't want to use, I think it was a question today about what the brain needs to use for energy, and it's not fatty acids for the most part. It's glucose, right?